Seed germination. Definition: we can define seed germination as seeds inside the fruits are designed to spread throughout the environment and grow into new plants. Is a process and is called as a seed germination. Types of seed germination. Seed germination is a fundamental process by which different plant species grow from single seed into a new plant. This process influences both yield of crop and quality of crop. During germination, the seeds take up water and results in swelling and softening of the seed coat at optimum temperature. Rupturing of seed coat helps to emerge radicle and plumule to form primary root and shoot. After emerging of radicle and plumule, seed activates its internal physiology and starts to respire and produce proteins and metabolize the stored food. This is final stage of seed germination, which brings out root and radicle out of the seed, and cotyledons are extended, which are the true leaves of new plant. Types of seed germination. We can categorize seed germination into three main types as hypogeal germination, epigeal germination, and viviparous germination. So, hypogeal germination. In this kind of germination, cotyledons do not come out of soil surface. It is called hypogeal germination. See here in this figure, this is a type where cotyledons, they remain inside the soil. They do not come out of the soil surface. In such seeds, epicotyle, that is part of embryonic axis between plumule and cotyledons. So this epicotyle elongates and pushes the plumule out of the soil. All monocotyledons show hypogeal germination, and from dicots, gram, pea, groundnut are some of the examples of hypogeal germination. So this diagrammatic representation represents the type that is hypogeal germination. In case of pea seed, this example will show here, and here you can see successive stages of hypogeal germination in case of monocotyledon. Now we will go towards epiphyseal type of germination. Here cotyledons are brought above the soil due to elongation of hypocotyle. Here this hypocotyle region, this one, it elongates and cotyledons are taken above the soil level, For example, castor, cotton or onion, you can see flat green lip-like cotyledons in the young seedlings. Here cotyledons, besides food storage, also perform photosynthesis till seedling becomes independent. In some other plants like bean, cotyledons being thick do not become lip-like. They shrivel and fall up after their food reserves are consumed by the seedling. So this is second type, and figure represents epiphyseal germination, where cotyledons are brought above the soil due to elongation of hypocotyle. The third type of seed germination is viviparous germination, also commonly called vivipary. And example of such germination is observed in mangrove plants. For example, rhizophora or Suneraceae or Heritera. So in such genus of mangrove plants, seeds cannot germinate on the ground, as there is excessive salt content in the soil and lack of oxygen in that marshy habitat soil. So in such plants, seeds dormancy is completely absent. See here example sketch of one of the mangrove. We can see, so there is no dormancy of the seed in such mangrove plants. Instead, embryo of the seed, when present inside the fruit, continues its growth while it is attached to the parent plant only. And this is called as a viviparous germination. So whenever on the mother plant itself, in the fruit, seed starts its growth. And this is a uniqueness of mangroves, and phenomenon is called as a vivipary. So here hypocotyle, this portion hypocotyle gets elongated and pushes the radical out of the seed, out of fruit. Growth of this hypocotyle continues till the hypocotyle and radical become several centimeters in length. Seedling becomes heavy now. As a result, it gets detached from fruit and falls down in the salt-rich, muddy water in such a position that plumule remains outside the saltish water while the tip of radical gets fixed in the mud. Thus, this protects the plumule. The radical quickly forms new roots and establishes the seedling as a new plant. So these are different methods or types of germination of seed. So while enlisting, we can enlist it as a hypogeal germination, second epigeal germination, and third viviparous germination. Now we will take survey of metabolic changes during seed germination. We have just now seen the things related to the germination, how seed germinate, and what are the types. Now actually when seed gets converted into seedling, during this phase of germination, what metabolic changes occur? So we will try to see the things related with these metabolic changes. Actually, life cycle of plant starts from seed germination and is a vital stage in plant development. Germination process begins with imbibition of water and passes through mobilization of food reserve, protein synthesis, and finally protrusion of radical and plumule. To sustain good seedling development, seeds store food materials mainly as protein, lipid, and carbohydrates. These are the sources of energy, carbon and nitrogen for seedling development. Hence, studies on metabolic changes during seed germination are important. So many changes, metabolic changes, physiological and biochemical changes occurs during seed germination. So what physiological changes, which biochemical changes takes place during seed germination, that we will study now. So related with physiological changes at first. So firstly, water uptake during physiological changes. What happens with water uptake, that we will see. So for seed germination, water is essential, moisture is essential. So water is absorbed by the seed through the porous seed coat or through the opening, whichever is there, through that water gets inside the seed and process of imbibition of water starts by the dry seed. Seed coat is due to imbibition of water, seed coat becomes more permeable to oxygen and water and less resistant to outward growth of embryo. After imbibition, the inner content of the seed increase in volume. Imbibant swells due to process of imbibition, and thus inner content of the seed increase in volume, and it exerts pressure on the seed coat, leading to rupture of the seed coat. The plumule and radicle gets emerged thereafter. So due to water uptake, what happens, what physiological change takes place that we have seen. So in the physiological change, next event, that is of respiration. So due to uptake of water and imbibition process, due to breaking down of the seed coat, the radicle and plumule, they start emerging out, and this is the initiation of germination process. So here the seedling requires, or cells require, enormous energy for various biochemical changes, and this energy is met through rapid increase in rate of respiration and the food reserve which is there in the cotyledons is used for this purpose, and particularly sucrose is the respiratory substrate at this stage, which is provided by the endosperm to the embryo. In oil seeds and pulses, the lipids and proteins respectively are converted into sucrose by suitable biochemical reactions. In third physiological change, that is mobilization of reserve materials. As germination process progresses, there is mobilization of reserve material to provide building block for development of embryo, to provide energy for biosynthetic processes, and also to provide nucleic acids for protein synthesis and embryonic development. So these are some of the physiological changes occur during the seed germination. Now, some of the biochemical changes. So related with the nucleic acids, in case of monocots, during imbibition, there is rapid decrease in DNA content and RNA content in the endosperm, with simultaneous increase in embryonic axis. High concentration of RNA in the embryonic axis precedes cell division. Due to more cell division, DNA content is increased. Another biochemical change related with the carbohydrate now. So, insoluble carbohydrates like starch are the important reserve food of cereals, endosperm. During germination, starch is hydrolyzed first into maltose in the presence of alpha amylase enzyme and beta amylase enzyme, and then maltose is converted into glucose by maltase enzyme. Glucose is further converted into soluble sucrose and transported to growing embryonic axis. During germination, embryonic axis secretes gibberellic acid, a type of hormone, into aleurone layer, which causes synthesis of alpha amylase. Biochemical change related to the lipids now. Many plants like castor, bean or peanut store large amount of lipids or fats as reserve food in seeds. During germination process, the fats are hydrolyzed into fatty acid and glycerol by the enzyme named as lipase. Fatty acids are further converted into acetyl coenzyme A by the process of beta oxidation. The acetyl coenzyme A is further converted into sucrose via Gluyas halide cycle and is transported to growing embryonic axis. Now biochemical change related to the proteins. Some plants have their reserve food in the form of proteins in their seeds. Here proteins are hydrolyzed into amino acids by enzyme named as peptidase. The amino acids may either provide energy by oxidation after deamination or may be utilized in the synthesis of new proteins. Then inorganic nutrients. So many inorganic nutrients like phosphate, calcium, magnesium and potassium are also stored in seeds in the form of phytin. These stored nutrients are liberated during germination due to activity of various phosphatases.